Show of hands, please.
Yes. So I had a doubt from the lab. I didn't
Yeah.
quite get like the gene ontology part.
Okay. Okay. Were you able to look at the gene ontology? Were you able to find gene ontology?
Yes, sir, but like I like the databases that I found, like there was a list of gene ontology terms, and like I'm not sure what to like what actually happens with them.
Okay. Okay. All right. So we can take that up. So the what I have the way that I have sort of designed the course earlier, I used to teach databases and their organization before I was teaching like going into these algorithms like the DNA patching matching algorithm. But then I was got a feedback from students that like it was too much of databases and it was kind of becoming boring. So the module just before uh, the minors is where I'll be covering these databases and all these doubts that you have about that you'll be collecting during the course of the labs will be cleared together in that module. So it's good that you are exposed to it. If you have a specific, I mean, if you have a specific question, then I can answer that specific question in Teams. Just type it out. And regarding the actual how ontologies are made and what ontologies actually represent, that is something that I'm going to cover anyways in class. Works for
Yes,
you?
sir. My doubt was yeah, my doubt was actually the broad picture itself. So
Okay, so
I guess I'll wait for
broad picture I'll give in the
yeah.
module. All right.
Sure, sir. Thank you.
Okay, so in the last class, I introduced you to a simple sort of algorithmic warm-up of finding uh, hidden patterns inside a DNA sequence. Specifically, we were looking for a hidden pattern in the origin of replication, and we found that the origin of replication is enriched for high number of 9 mers, origin of replication in Vibrio coli, a specific bacteria, 9 mers, 3 9 mers, and 3 reverse complement of those 9 mers, which actually represents what's called DNA boxes. And it is very surprising to find 6 or more 9 mers within a um, short span of 500 nucleotides. So here I gave you 2 homework problems. I hope you have worked on these. Does anybody know the answer?
Sir, I have done, but like just by guessing. Sir, can I tell you the answer? Then you can tell if it is correct or not.
Okay. I mean, it's a simple combinatorics problem, right? So I don't understand what, like, there is no intuition involved here. But please go ahead.
I didn't spend any much time. I just written equation on my own.
Huh, so you will write it on your own only, na? That is what a homework problem means, no? So yeah, tell me the answer.
Okay, so sir, first answer will be um, 492 upon 4 to the power 9 plus 8 into 6. And the second will be n minus
4 to the power 9
plus 8
plus 8.
and the whole into 6.
This whole into 6.
Yeah, in
Can
yeah,
can probability be larger
denominator.
than 1?
What?
Can probability be larger than 1?
No, never.
So
Oh,
your
I'm
answer.
not saying that it is larger than 1.
So what is the your answer is larger than 1, no?
No, it is not. Oh, I told it. I did. Okay, sir. I will calculate and after that I will tell you.
Okay. Okay. So yeah, I mean, this kind of problems you can expect in exams, as I said. All right. So now that we have figured out how to look for hidden messages in Vibrio coli, uh, can we just quickly apply the same understanding to other bacteria to find their origin of replication? So if we'll go to another bacteria, Thermophila petrophila, which basically is a Thermoticus petrophila. This is basically a bacteria that loves to grow in heat, and it's an extremophile. So what we find is that there are no particular occurrences of the DNA A boxes that we had found earlier, right? So these sequences, ATG, ATC, AAG, and its reverse complement are not found here, right? So does this mean that this does not have like this does not have DNA binding sites? So in fact, if we do apply the algorithm, the frequent word algorithm in the ORI, we actually find that there are other frequent words which are found here. Uh, and particularly that essentially means that if you have a different genome, that means there is a different hidden message. So initially, when I was asking you about trying to find the origin of replication, you can find an origin of replication. And somebody said that, okay, if we know the exact locations where the origin of replication is sort of found, we can just search for that sequence. So from this example, it basically shows that if you have a different genome, you are going to have different hidden messages or different DNA box binding boxes in different organisms. So therefore, in order to find the origin of replication or these DNA box binding sites, you have to run this algorithm for every new species again. All right. But we have solved not the complete problem. So this is what this is the frequent word, right? GTC, GGC, and this one, which is actually reverse complement. And sorry, not this one, and this one are reverse complements, and this is what this is what we find as the DNA binding sites. So now if we actually look at this idea, does this actually give us, can we find the origin of replication now based on this idea? So we have found there were two problems, right? One problem was what are the hidden messages within the ORI where the DNA box could bind and begin replication. That was the first problem, right? So we have found hidden messages if the ORI is given, but how do we find actually where is the ORI, right? Now that we know that hidden messages may differ, how could you look for the ORI in a newly sequenced bacterial genome? So if I give you a newly sequenced bacterial genome, a lot of people in our department actually are discovering newly sequenced bacteria. For example, Professor Preeti works on discovering bacteria that are eating up plastics or petrol. Professor Lucinda is discovering different bacteria which are conducting electricity on their from their membrane, right? So like that, a lot of new bacteria are being discovered. So if you are given a totally new bacteria, how is it that you would actually find that window of 500 base pairs where the ORI actually lies, or where to look for different frequent words? Okay, so the old strategy was that given a previously known ORI, around approximately a 500 nucleotide window, find frequent words, right? Or clumps, as you call them, as we can call them in a ORI. And these will become candidate DNA boxes. All right. So the origin of replication is going to have lot of frequent words, right? So can we use this strategy to look for the origin of replication in a genome? Can somebody tell me? How can you use this strategy?
We cannot use it like there may be lots of other frequent words which doesn't indicate ORI. Like that happens in case of like human genome. We have lots of satellite DNA and other kinds of microsatellite, mini satellite DNA. So it is not possible to say that just by frequent words will say that here is the ORI.
Okay, that's good. So basically, the strategy would be um that you will take this window, right, of 500 base pairs, and you will move this window along the genome, and you will ask whether there is existence of any clumps or any frequent words in these windows, which is the probability of finding which is very rare. So if it's a rare event, that means that event is probably meaningful, right? So we take a window and we go from one end of the genome and keep on moving, right? And then we find, okay, and there are some windows where there is higher frequency of uh, these clumps, or there is high frequency of frequent words, right? So the new strategy is find all words in all frequent words in all windows within a 3 million nucleotide genome, right? So now basically the windows with clumps of frequent words can be candidate replicates of candidate regions of replication of origin, right? So the exercise now is to define a computational problem modeling our new strategy. So the computational problem then becomes defining and hunting for clumps. A Kamer here forms a clump L comma T, right? So we define a clump as L comma T inside a genome. If there is a short interval of length L in the genome, which has at least appearance of a frequent word at least T times. Okay, is this clear to everybody? So we call a clump a clump. Basically, you do need to have a window of length L, and that window should have at least T occurrences of a Kamer, right? So Kamer can here be K could be 9, K could be 8, K could be 10. So in the case of DNA boxes, K is 9. So computationally, if you were to define this problem, the clump finding problem becomes the input is a string, the genome. Then you have integers k, which basically tells you the length of the DNA A box binding site, the length of the a window L. So L defines the probable region in which you are going to find this clump of or this high occurrence of frequent words of length 9 or length k, and T is the number of patterns in such a clump. All right, and the output would be all k-mers forming L comma T clumps in the genome. Is this clear?
Yes, sir.
Yes, sir.
Okay. All right, so we go for the pseudocode. You have your input as your text. So this text is nothing but the genome. K is length of your k-mer. L is the length of the window. T is the number of occurrences, least number of occurrences that should be there for the k-mer. And there is something called your H that we'll talk about. So patterns, again,
What is
you
age,
don't
sir?
know what. Say that again.
What is age?
We'll talk about it.
Okay, okay.
So you go, you basically now first thing that you do is you extract a window. So you extract a window from the beginning of your genome, right? From 0 all the way to n minus 1, n minus L, right? Because it's the same thing. If you're trying to extract k-mers, it will be from 0 to n minus k, where n is the length of the string. Similarly, you are extracting windows, which has the same idea. Then for each window, you apply the frequency map, right? And you get all the most frequent words. And now for every string pattern and frequency map, you ask if this pattern and frequency map was occurring more than t number of times, then this is a pattern which is worth remembering, right? And then in the end, in this case, you will have to remove duplicates because two windows can have the same frequent word patterns. Okay, but there is a complication. Complicated functions can be made easy. Remember, with the frequency maps and routines. All right. So uh, as I asked you, if you specifically look for bacterial genomes as a source of hidden messages, this is destined to fail because if you would have solved the homework problem, you will find that the probability of finding such clumps of finding nine mers is finite, and it's not very small. It is small, but if you multiply by the number of potential windows that you are going to see, so if in a 3 million base pair genome, you are going to see 3 million minus 500, so approximately 3 million windows you are going to see. And if you multiply the probability of finding a clump in a single window by 3 million, this probability becomes quite high. What does this mean? This essentially means that there is an issue of repeats. So, for example, Ratnesh was mentioning microsatellite DNA and other DNA. There is one kind of DNA, another kind of DNA which is there in humans called ALU repeats. A lot of work for ALU is actually was done in India, very close to IIT. just about 10 kilometers away from IIT. And just like ALU elements, so these are about 300 base pair fragments which are occurring lots of times in the genome. So just like that, even in bacterial genomes, there are many repeated chamers which are appearing, which might actually lead to some spurious windows that might be, you might say that, okay, this window has high frequency of 9 mers, but that could actually be because of a lot of that particular 9mer being very often seen in the bacteria, right? Because of issues of non-random distribution of repeat regions. Does anybody know in the class here why do repeats occur? Do you have a guess? Can you please be louder?
Sir, the non-coding
Because of
DNA.
recombination.
Okay. So first, I think Deepak has raised his hand. Yes, Deepak.
Sir, I guess the non-coding DNA have many repeated sequence, so they are just like used to just maintain the gene sequence into the chromosome, and intron sequences are particularly repeated again and again.
But why do they have repeat? Like the question is not, I mean, it's an observation that they have repeats, but why do you think they have repeats?
Because sir, they are just to just maintain the genomic structures, so therefore their means their work is to just maintain the genomic structure. They have no functional use, so they have repeated sequence.
Okay, so there is no evolutionary pressure because they are not making a protein. So
Yes
they
sir.
don't have to necessarily have the codons in place. So if you look at the codon table, most codons actually are not coming from repeated nucleotides, right? So if you have AAA, UUU, GGG, CCC, those are not often the most preferred codons in the cell. That's one. That's good. Could you think about the reason why repeats could be uh, biochemically more, like could be biochemically present inside a cell? If you go back to how DNA replication happens.
Yes sir, they are important because in case of the plasmid replication, we need some repetitions to terminate the replication. So in that case, they are useful.
Okay. Anybody else wants to give it a try? Deepak, please keep your hand down. Ratnesh, now you want to give it a try?
No, sir.
You don't want to give it. You had something, right? You had the same idea or?
Sir, I told that repetitions are there because of recombination, and that happened once at a time, and after that, uh, if those repetitions are not causing any kind of metabolic burden to the cell, they are just there and they are not doing anything, and just replicating from generations to generation, and being passed on. They don't have any use, and they are just as they are there. They are there.
Okay, so the combination, maybe that's not the only reason, but yeah, there's another reason, something called DNA slippage, because whenever you have a homopolymer stretch, so whenever you have a lot of nucleotides, AAA, TTT, right? Then when the DNA polymerase is going over this, the replication is happening. Sometimes it slips. So sometimes there are more A's or more T's incorporated in the strand because the DNA polymerase slips. So this is also one of the reasons why this happens. All right. So now, if this are this this kind of implementation fails, right? If you actually apply this hunting for clumps function, then this fails because bacterial genomes or most genomes tend to have lot of clumps of such frequent k-mers, right? So now can we use some other information that we could actually uh, that can help us to pinpoint where the origin of replication might be there. So in the case of E. coli, for example, there are approximately 2000 different 9-mers that can form these 500,3 clumps. So it's probably unclear where the ORI points to. All right, so we are back to where we started initially in the last class. The biologist will say, let's just hack out every piece of DNA that we can do and see which pieces are required for the genomes to replicate. And correspondingly, you can, the biologist will essentially say, I need more information to hack this problem. All right. So now we go back to the biochemistry and try to understand, is there something that could, something in the process of DNA replication that could help us to come up with another algorithm to solve this problem. So now there is a surprising pattern if you look at the nucleotide counts themselves if you walk along the genome. Okay, so a very simple computational analysis one can do. One can go from the beginning of a genome till the end of a genome, right? And you can calculate the frequency of nucleotides in a specific window. So let us say you take windows of 500 base pairs like we were doing earlier to find clumps, and we move windows of 500 base pairs from left to the right, and in each 500 base pair window you calculate the frequency of Cs. Okay, so now what we see is that for half of the genome the frequency of Cs is more than 25%, and for the other half of the genome the frequency of Cs is less than 25%, right? So this is quite surprising, and this seems to happen. Can somebody tell me why is the expected frequency of C is 25%?
Because of
So because it should be
80 GC four
equally
base
probable, right?
Yeah.
four bases are there.
Okay, good. So because there are 4 bases, so it's 1 in 4. So the expectation is if there was random distribution of ATGC, then all of them should be present at 25% in each window, right? But this is surprising. So the frequency of C increases and then it decreases. Similarly, if you look at frequency of G, the frequency of G is decreases first, and the frequency of G increases later on. All right. Can somebody tell me why is this the case? Why would
Because
the
G
frequency
and
of
C
G and
are complementary to each other. So
okay?
when the C will be increasing, G will be okay. Okay, so sir, I don't know. Like if both should increase and both should decrease at the same time.
So this
Is
is
that the
on one
case?
single strand or both the strands?
You usually
So,
are looking at only a single strand.
sir, as they are reverse complementary, so on the different side they should increase and decrease.
Are you sure about that? So if I just have a DNA fragment, right, which is A, this has no G's or C's.
So I
Or
would say
if I
because like the overall GC content of a single strand should be between 40 to 60%. So on an average, it should be around 50 to 55%.
nobody says
So
that.
that is why.
There is no restriction on the average GC content. You can have less GC content. Malaria, for example, the malarial parasite P. falciparum that has extremely low GC content, that is mostly an AT-rich genome. Approximately 60 to 70% of the genome is just A's and T's. Nobody stops that parasite to have less G's or C's. There is no bound like that. So this is an observation, okay? So this is an observation, a surprising observation that computationally people came up with, okay? And now let's try to understand what this is, why this happens, and can we actually use this to find the origin of replication.
Like in case of you were saying that of 40 to 60, 60 to 70% are only A and T's. So isn't it that they don't how much they have conserved sequences? Like all organisms must have some conserved sequences, and in bacteria also maintaining that a species conserved sequences must be there. Like
This is an observation. So they have probably conservation within the parasites, but if you compare the parasite P. falciparum with a bacterial species, so there are different species, they are millions of years apart. So they have enough time to accumulate differences which are more relevant to their species than a bacterial species. Okay, so now when we take the difference between the frequency of G and frequency of C across these windows, we see a extremely stark pattern that almost at the half of the genome is having low G minus C frequency and half of the genome has high G minus C frequency. Okay. So now if this pattern is still there, if you actually look at the pattern, this pattern where the G and C frequency actually changes dramatically is where you have either your origin of replication or termination, where the replication terminates located. All right. And this is something which is across an arbitrary position in the genome as well, because I mean, a genome you can just take, because it's a circular genome, right? So you can basically take bases from here and you can move here. The genome is still circular, so it doesn't matter. The origin of replication is going to be still located at this location, and termination is going to be located at this location. All right. So this G and C difference seems to be playing a very interesting role in marking where the origin of replication is happening. And we are trying to now understand by going back to how DNA replication is happening.
I had a question.
Yeah.
So is this the case for every organism or is this the case for specific organisms?
This is the case for mostly bacteria. For higher organisms, things become more complicated as expected. But the whole goal is here to explain you that there are certain, if we go back to how things work, we can explain a lot of I mean, we can come up with algorithms. If we understand how molecular biology work, we can come up with algorithms to find uh, locations like the ori. I'll come to how things work in other organisms specifically.
Fine.
Okay.
So basically, like this is saying that the region between the origin and the termination is mostly C-rich in bacteria.
You look at it. There's a very interesting revelation that you will have at the end of this class, hopefully. So this is how a DNA strand looks like, right? Double-stranded DNA of a bacteria, it's circular, right? And let us say the origin of replication is shown here and the termination is shown here. So ori is represented as ori and termination is represented as ter C. The strand which is going in the clockwise direction, that is 5' to 3' is shown in blue. The strand which is going in the counterclockwise direction at the bottom, which is a reverse complement to it, is shown in green. Okay. So now when DNA replication has to start, can somebody quickly tell me what happens first? What structure do you see appearing on DNA when replication starts?
So a replication bubble.
First of all,
You see a
replication
replication
bubble.
bubble. Exactly. So the DNA is going to get opened up, and because of which, when the DNA gets opened up, you have this kind of a bubble come up. All right. So now when this DNA opens up and the bubble comes up, now the DNA polymerases, specifically 4 DNA polymerases, can do this job of replicating these opened up bubble, right? So now let's look at the models of how DNA polymerases can work. So either you can have 4 DNA polymerases, right? Where one DNA polymerase goes in this direction, other DNA polymerase goes in this direction, the third one goes in this direction, fourth one goes in this direction. Is this correct?
No, sir.
Why is this not correct?
Because DNA polymerases can add bases only in 5' to 3' direction. They
Yes.
can only add to remove OH and add that a basis.
In the phosphodiester bond. Very good. Everybody agrees
Yes,
with
sir.
this?
Yes, sir.
Everybody in the class, I hope this is some very basic molecular biology that I hope even if you are sitting in a board of a company as a startup founder, that if somebody asks you what's the direction of DNA polymerization, it's 5' to 3', right? I hope everybody is on board
Yes,
with
sir.
this. Okay, good.
Yes, sir.
So this is actually not how it happens because DNA replication happens 5' to 3', right? So this is wrong. So how does it actually happen? DNA replication is actually unidirectional. And if it is unidirectional, certain interesting phenomena starts to appear. So first thing is, let us say the DNA replication starts in this direction. So this is called the leading strand because it starts to replicate, and as the DNA replication bubble will open, this DNA polymerase is going to keep on sitting and keep on doing the replication, right? Similarly, on the other half of the bubble, another DNA polymerase is going to sit, and as the DNA replication bubble opens, it's going to keep on replicating, right? So this specific part here on the top becomes leading half strand, and this specific green part here becomes the other leading half strand, right? So typically whenever you are taught in your molecular biology class, you will be only talked about half of the replication bubble, right? But this is actually how the real replication bubble looks like. You have two leading strands when you are talking about the replication bubble. Now, on the other strand, you know about Okazaki fragments. As and when the replication bubble opens, a DNA polymerase is going to sit and is going to replicate the exposed part of the DNA, and this will form multiple fragments. Okay, so this particular strand which is going to be replicated like this is called the lagging strand. Okay, so when the fork opens, then you have these small fragments come up. When the fork enlarges, you will have another of these small fragments come up, right? And so on and so forth. So at the end, and these fragments are called Okazaki fragments. And at the end, what you're going to be having, you're going to have the leading strand replicated completely in both directions, and you're going to have multiple Okazaki fragments in the other half of the lagging strands, two lagging strands. All right. And at the end, what happens is there is an enzyme called DNA ligase, which comes and ligates these DNA bridges where no replication, where basically a new DNA polymerase came and sat and completes this into a single genome, right? That the genomes are replicated. Now, because this thing happens, some interesting phenomena begin to emerge. The leading strand often spends a long time as a single stranded DNA molecule. All right. So the lagging strand, the lagging strand. So the leading strand typically is not exposed. As soon as the DNA polymerase is sitting there, the leading strand basically is always double stranded. So as soon as this opens up, the fork opens up, the DNA polymerase will move, and it will basically is going to form the reverse complement strand of the leading strand. But on the other hand, the lagging strand is waiting for the DNA polymerase to come and sit and initiate the polymerization process, because the first time the DNA polymerase comes and binds to the DNA, that's what's called the rate limiting step. So that takes some time. On the other hand, if the DNA polymerase is already sitting, then it just keeps on going and it almost unzips. The process of unzipping is also facilitated by the polymerization process. I hope everybody is clear with this. Can I hear a yes, please?
Yes.
Yes, sir.
All right. So now when this happens, because the lagging strand is waiting, so now, I mean, why would you care as a computer scientist? So when the single stranded DNA, when it's waiting, it has a much higher mutation rate than double stranded DNA. All right. So what does it mean? That if one nucleotide is having a greater mutation rate, then we should observe the shortage of that particular nucleotide on the lagging strand. Okay. Do you know which nucleotide has a higher mutation rate than other nucleotides? Can you guess after looking at the plots that I showed you?
No, sir, I cannot.
Do you remember the structure of nucleotides?
Would C have a higher mutation rate?
Why would you say C would have a higher mutation rate?
So because of the plots that we saw earlier.
You're
I was
guessing. But do you know about
yes.
the biochemistry? Can you think about the biochemistry? Quickly go. It's not an SN1, SN2 reaction. So you're not in 11th, 12th class
Not
preparing for
triple
GE.
bond, so maybe something along those lines.
Exactly. So the C essentially can rapidly mutate to thiamine because it can very quickly go into a deamination reaction, and deamination reaction specifically is 100-fold more faster in single-stranded DNA than a double-stranded DNA. So what does this mean? When you have a lagging strand, that is, if you have a C on the lagging strand, then this C is quickly going to get mutated into a T, right? And the leading strand is going to maintain what was there on this on this strand, right? So now when you keep on doing this over multiple generations of um, replication events, even within a single E. coli's life, it's going to replicate 50 to 100 times, and you would see you will be making a lot of lagging strands. So basically, even at the end of lifetime of one E. coli, a single E. coli, the original one in your culture, it's going to accumulate mutations in the lagging strand more often than the leading strand, and those mutations are going to be more often occurring in C rather than any other base. Okay, so now when you walk along the genome, you would see that in the direction of your in the direction of your leading strand, you are going to have a high C and a low G. Okay, so the G minus C is going to be decreasing as you walk along the leading strand, leading half strand basically, and the G minus C is going to be increasing as you walk along the lagging half strand. So G minus C is going to be decreasing in this direction, and G minus C is going to be increasing in this direction. Okay, so you can walk along the genome, you can look at the frequency or the value of G minus C in a window, and if it suddenly changes from decreasing to increasing, then you most likely are at the origin of replication. So it's decreasing, 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 and suddenly it starts to increase. It's increasing, 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 and suddenly it starts to decrease. So you are at termination when you are having this kind of phenomena, and you are at origin when you have the other kind of phenomena. All right. So can now we use this information to pinpoint where the origin of replication might be lying? Yes, no?
Yes, sir.
Yes.
Computationally?
Yes, sir.
So in order to solve this problem, we make what's called skew diagrams. So a skew diagram can be defined. Skew for nucleotides K is equal to the number of Gs minus number of Cs for the first K nucleotides of the genome. All right. So if you are having skew at till 5, that means if this is the plot shown here. So at the first location, G minus C is going to be minus 1. Then for first 2, it's still going to be minus 1. First 3. Then a G comes, so it increases. Then another G comes, it increases, 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 right? So that's how a skew diagram is essentially made. So now if I were to ask you to write uh, a code to calculate the skew diagram, I hope you can do that.
Yes, sir.
Okay.
Yes, sir.
So now what will the skew diagram of a bacterial genome look like? So it typically looks something like this. You have increasing, then it decreases, then it increases again. Now going back to what I told you, can you point out where the origin of replication will lie?
Where G minus C SQ will be 0.
Somebody else wants to give it a try?
So where we have like the
Here
sharp
you see
peaks or
zero.
the sharp dip.
So here or here?
So probably the upper one.
So I'm showing you the figure again.
So I guess it would be at peaks because at peak, before the peak, the G minus C is increasing, and after the peak, G minus C is decreasing. So it would be at peaks.
So it was decreasing and then it immediately increases. It was decreasing, then it immediately increases. This is where the ora is. And correspondingly half across, typically you have your termination. Okay. So now we have solved the first question. you first have to solve two problems. You have to solve the skew problem, where you have the skew changing, the location where the skew is changing. You have to extract the window there, and you have to look for
So one more
frequent
question. The
ones.
skew diagram is also like 5 prime to 3 prime, right? Yeah.
Yes.
Always. Okay.
Always you will have the DNA sequence is always presented in a 5 prime to 3 prime direction. That's why that's the case. This is the case.
Here in
Everybody
circular
understood?
DNA, what is the meaning of 5 prime to 3 prime?
Say that again.
In circular DNA, what is the meaning of 5 prime to 3 prime? How will determine like whether it is 5 prime to 3 prime? Looking at the chromosome and not looking at the data. Like
So looking at the chromosome, you cannot say it. But when you look at the data itself, the way that sequencing happens, you know that the sequence that you have obtained is a 5 prime to 3 prime sequence.
but how will we say that this is 5 prime to 3 prime? Like we can sequence in any way, like in one direction or the reverse direction.
So I think when you have the genome sequenced, one strand is taken as the 5 prime strand, and the other strand is taken as the 3 prime strand. So this is basically, I think it is based on the number of genes. The strand which has more number of genes is taken as the 5 prime strand, as the Watson strand.
Yes, sir, probably. Yes.
So this is an arbitrary thing. So what your question totally makes sense. You can sequence from both the strands, right? There is no strand specific kind of sequencing technique. I mean, there is, but typically what people do is when they are having sequencing, they assign one strand to the Watson strand or positive strand, and the other strand as the counter positive strand. And whenever you have these genomes represented, they are always represented from 5 prime to the 3 prime end for the strands that you have sequenced, or for the direction that you have chosen to be sequenced, chosen as sequencing. Because if you would be representing the same information, right, in the reverse complement fashion and sequencing in the reverse complement fashion, then you would also be showing the reverse complement. Okay. So the plot doesn't change. Your question is with respect to the plot, right? Right, Ratnesh?
Yes, sir. Yes, sir.
So even if if you have, say, arbitrary, you add the other strand as the reverse strand, then the diagram will not change because the reverse complement will be present. So it will be complemented and then things will be reversed. So every G is going to be replaced by a C. Okay. So do
So
it, do it.
we are so sir, we are not running that 500 window on entire genome sequence. We just directly calculate
I'll show
the
you. I'll show you.
okay, sir.
First thing that you do is you first try to run this skew diagram. You locate exactly where the ori is changing, right? Where this kind of phenomena is happening. You extract the 500 base pair window here. And in this 500 base pair window, you run the frequent words
Approach.
subroutine.
Okay, sir.
Okay.
Okay.
So you have the minimum skew problem. You've been given an input DNA genome string, and you find the minimum value of the skew. And at the minimum value, typically you are going to have the ori present. So this is what was actually used not very long ago in 1998, where using these cumulative skew diagrams. So the single author paper, he basically was able to assign origin of replications to lot of bacterial genomes. And this is actually how, if you would have any bacterial genome nowadays, that you will be essentially, that's the first step of assigning the origin of replication. Okay. So but if you look at E. coli specifically, there is no frequent 9-mer that appears more than 3 or more times. Okay, so this is a problem. Should we give up? Do you have any ideas?
In every part there won't be origin of replication. Like in genome of bacteria, everywhere not the origin of replication.
So I'm saying that in
this
E.
part
coli,
not be not have.
no, in E. coli, at the origin of replication, we found the origin of replication in E. coli using the skew diagram. But in that origin of replication, there are no frequent 9-mers.
So is it compulsory to have a 9-mer? I mean, we
So
could
we
look
can
for 8.
reduce the substrate length, right?
Okay. So the substrate length could be different. Any other ideas? I had talked about it in the previous class, previous lecture. Could there be certain mutations coming up just by chance to make the binding a little bit more promiscuous? Right? So actually you do see that you have 9-mers, but these 9-mers have one mismatch between them. So you have two 9-mers coming up, but they have, for example, this is TGT here, but the other one is TTT. So there is like one mismatch that that occurs. Okay, so big with this mismatch, essentially, you have the DNA A box is sort of promiscuous. The protein is not looking for an exact match; it's looking for a almost match. All right. So just like that, we have solved the problem. But now if you want to apply the problem to all genomes, to all organisms, there are obviously going to be complications, right? What are the complications? Some bacteria have fewer DNA boxes. Okay. Sometimes the terminus of replication is not located directly opposite to the ori. The example that I showed you here, if this is the terminus of origin, this is the origin, then this is the terminus of replication based on this understanding of the skew diagram. Okay. But when you look at the genomes, this is not always the case. So if we go back to this Thermophilus petrophila, here you do see a low point, which is where you'll have the ori, but there is not particular very high point uh, of the GC bias. Okay. So the moral of the first problem that we have tried to solve is that there is no appropriate division of how biology in general should operate. You have some information that you gather from how processes happen, how replication is happening, and then you can use these ideas in order to define computational problems to solve come up with the solution of these problems. Okay. Now, a homework problem. I gave you homework problem earlier as well. Remember these are some problems which are telling you what are the kind of questions that could be asked. If this is a sequence and this is the location of origin of replication, and assume that in each cycle of origin or in cycle of replication, 50% of all C's in the lagging strand are getting mutated. So if you can quickly look at this sequence, you will notice that it is ATGC, 4 bases repeated again and again. If you were to mutate, if you were to essentially do 3 rounds of replication, you would already see how the leading strand and the lagging strand, they have a different GC content. Okay. So 50% rate of replication, rate of mutation is very high, but imagine that we are just speeding up evolution. So typically this rate of mutation is going to be one in a million, right? So we are making it a million times faster, right? So million times evolutionary years. And yeah, so you will clearly start to see how the GC bias accumulates when you solve this problem. The other problem, just because we sort of missed one lab in the confusion of when to have the lab, is to actually go to this tool given here and see how an ORI finder works. So basically you can, using the platform that in the lab we saw last time, the NCBI platform, take a bacteria of your choice, right? Maybe you can take a bacteria which was the first bacteria that was sequenced. Can somebody tell me what was the first bacteria that was sequenced?
So
Something that,
Hemophilus
Philus.
influenza.
yes, Haemophilus. So maybe you can take Haemophilus influenza's bacterial genome from NCBI and you can stick it here and you can run it and find out uh, where the origin of replication lies. And once you have figured, like use this ORI finder, what I would like you to do is try to actually come up with a pseudocode, uh, write your own pseudocode to implement the ORI finder tool. This is very important because I can guarantee you there'll be at least one question from such pseudocodes that I'm asking you to write in the lab. So coming back to the question that I think Vishantam, you had the question about does it happen? Is this what happens in all the species? So we know that bacteria has a single origin of replication, right? So you can have this idea for organisms with single origin of replication. But if you go to higher eukaryotes, which don't have circular genomes, you have multiple origins of replication.
Yes, sir.
Right? So now the problem there is not to find a single origin of replication, but to find multiple origins of replication. So applying this idea, you can essentially walk along the genome and can look and can find multiple places, local minimas, where it goes from low to high, where you could be accumulating such mutations. Right? So if the problem
But
is to
like,
find, yeah,
since we don't have a circular genome, then like we won't have as much like that similar problem of C mutations, right? Because like it shouldn't be as prevalent as in like probably circular genomes.
why would you say so? The lagging strand is still going to be there. Replication
So
is
it's
still going
going
to happen.
it's going to be there, but like maybe I'll have to look into it a bit more from my own
Yeah,
end.
I mean, uh, if you do the homework problem that I've given here, right? When I'm asking you to do this, this is not assuming any circularity, right? It is just based on where the aura is lying, right? If the
Yes.
aura is lying here, so aura itself, the location of aura is determining how the GC content is distributed across the aura, not the fact that these things are actually circular or not.
So one more thing, for example, if we have say 10 different origins of replication, so like the repeating pattern or not the repeating pattern, yeah, or the pattern which is prevalent more in the ORI, would it be same for all the ORIs or can it be different for different ORIs in the same genome?
In when you're talking about a eukaryotic genome with multiple origins of replication.
Yes.
So typically you can already see that E. coli has this kind of promiscuous DNA binding sites. So in eukaryotes, the promiscuity is usually higher. So you're going to have the patterns of DNA boxes, the clumps is going to be a slightly more complicated. There are going to be more errors or mutations that would be tolerated because you would want to have the same protein bind to many different places. And if you are dependent only upon a specific sequence, then it's possible that just by chance, if a mutation happens, hits that particular base, it might actually lead to the replication not happening in the cell.
Right.
Okay, so I'll stop the class here. If there are any questions, I can take them. Otherwise, you're free to leave for the next